Okay, let's talk about cellular respiration. So this is the overall diagram for cellular respiration. I'm going to go back to this initial diagram that we talked about with the mitochondria in it. So in the mitochondria, you know that we have this, and I'm going to kind of go over to this like little cross-section area. So you have the outer membrane, you have the intermembrane space, which is the space in between the two membranes, and then you have the inner membrane. So that's going to be majorly important when we start to go through these diagrams. So the first process involved in cellular respiration, and this happens in both plants and animals, happens in the cytoplasm of the cell. So not necessarily in the mitochondria or the chloroplast, it's happening outside of the mitochondria in the chloroplast, in the cytoplasm. So that process is called glycolysis. So glycolysis is actually the process that we're going to split glucose into two parts. So I kind of gave this to you as its own separate diagram. So glycolysis is going to happen in the cytoplasm. You're going to start with glucose that you know that you get that from eating different foods. So glucose is going to diffuse into the cell. This is your chemical formula for glucose. What's going to start to happen is we're going to start to break down glucose, because remember, the process of cellular respiration is all about breaking down glucose to create energy in the form of ATP. That's our overall goal. So this first part of glucose is our anaerobic part of glucose, meaning there's no oxygen anywhere in this diagram if you take a look. So the first thing, we're going to split glucose. We're going to use 2 ATP. So this is where it gets funky. We actually have to use energy to get energy. It's kind of like when you start a fire. In order to get a fire started, you have to put some energy into it in order to get it going. But the idea is that we're going to get more energy out than we put in. So we're going to start by using 2 ATP. We're going to use the energy and take away a phosphate and convert it to 2 ADP. So we use that energy to start it. From there, we break glucose into 2 G3P molecules. So 2 G3P molecules, which if you remember from photosynthesis, that was basically half a glucose. So we split it into 2 pieces. Then what happens is we start to then take away some hydrogens. So where in photosynthesis we had NADP, because they were involved in photosynthesis, we now have NAD molecules. So the NAD molecules are going to pick up some hydrogens. So we're going to strip some hydrogens away from those G3P molecules. You're going to take 2 NADs. They're going to pick up those hydrogens. They're going to drop them off at the mitochondria. They're going to come back up to get more. So again, NAD is just like a taxi. It's going to keep going on this little trail back and forth. It's going to pick up its hydrogens. It's going to drop them off. It's going to go back and get more hydrogens. So we get rid of hydrogen. Anytime we break bonds, we're going to release energy. So by breaking off hydrogens, we have given ourselves the opportunity to create some energy and to store some energy. So we're going to take 4 ADP molecules now. We're going backwards. We're going to hook on a phosphate and we're going to form 4 ATP molecules. So we start with glucose. We add in some energy, make it unstable. It forms 2 G3P. We take away hydrogen, we take away energy, and we end up with what we call 2 pyruvate molecules. Those pyruvate molecules are then going to go into the matrix of the mitochondria, which is the middle section of the mitochondria. So when we talk about our overall process of glycolysis, again, it is anaerobic because there was no oxygen. We have a net gain of 2 ATP because you're probably saying to yourself, but wait, I thought you just said we formed 4 ATP. Well, when we talk about our net gain, we're talking about an overall gain of energy. Because we had to spend 2, we only gain 2 ATP. How many total are produced is 4, but we only really, at the end of the day, are going to gain 2 because we had to spend 2 in the beginning. So this is the process of glycolysis. So then from here, and this is kind of our overall diagram, this is happening outside of the mitochondria, again, in the cytoplasm. So we show our hydrogens that they come up from this cytoplasm through the outer mitochondrial membrane, and they kind of form in this space. Your pyruvate is actually going to diffuse all the way into the matrix of the mitochondria for what we call Krebs cycle. So this is it looks similar to Calvin's cycle that took place in photosynthesis, but it's not really. So I'm going to walk you through, I'm going to follow the pyruvates now. So your pyruvates are formed in glycolysis. They diffuse through the outer membrane, through the intermembrane space, through the inner membrane, into the matrix of the mitochondria. In the Krebs cycle, you start with 2 pyruvates. Again, we have this NAD molecule, so NAD is going to pick up some hydrogens. 10 NADs are going to take their 10 NADH2s up to this inner mitochondrial membrane. So the inner mitochondrial membrane is where this electron and hydrogens are basically going to get dropped off. So this is going to look familiar, and I'm going to come back to this in just a second. So you're going to strip away some hydrogens. You're going to strip away 6 CO2, which, if you remember the overall process for cellular respiration, 6 CO2 is a product, which you know it diffuses out of the cells and then you exhale it as waste. From there, because again, we're breaking energy, so we're going to be able to store some energy, we are going to form 2 ATPs. So the overall process for Krebs cycle, which is step 2 in cellular respiration, happens in the matrix. 2 pyruvates lose some hydrogens. NAD is going to take those hydrogens to the inner membrane. You're going to lose your 6 CO2. You're going to form 2 ATP. So your gain of energy from Krebs cycle is 2 ATP and 10 NADH2 molecules. So the next part of the process is the electron transport chain. So basically, as your NADH2 brings up, whoa, sorry, got a little out of focus there. As your NAD molecules bring your hydrogens up, this is called the electron transport chain. It's found in the inner mitochondrial membrane. So NAD drops off its hydrogens, and the hydrogens donate their electrons to these receptor molecules that we call the electron transport chain. So essentially, similar to what we saw in our photosystems, it's kind of similar to that. So our electron kind of gets transferred from one of these molecules to the other, and you're seeing this AT right here means active transport. So then what we're going to do is that hydrogen that donated its electron is now an ion because it lost an electron. It has a positive charge. So it's basically a proton, right? Because a hydrogen ion is just a proton. So it's going to pump, that energy of that electron is going to pump that hydrogen into this intermembrane space. So basically what the electron transport chain is helping us do is it's helping us take these hydrogens that came from our pyruvate, and they're pumping them into this interspace. So we're building a gradient here. We're using energy. We're actively transporting these hydrogens into this space. We also have some coming from glycolysis, so we're building up a gradient in this space. So once we take this energy away from this electron, it gets kicked out into this matrix again. So we basically take an electron, we pump it through this system, and we kick it back out. But what that electron is going to do is it's going to help us with this next process. So step 1 was glycolysis, step 2 is Krebs cycle, step 3 is the electron transport chain, which the point of that is to build this gradient of hydrogen ions, because again, you're going to see this ATP synthase molecule that you saw in photosynthesis. So the hydrogen ions are going to be forced to come through here because we've built a gradient, right? They want to go from high to low concentration. So as they come through, and there will be 32 of them total that come through, you're going to charge up 32 ADPs to form 32 ATPs. So there's your big energy bank, right? That hydrogen ion is then going to hook up with this electron that got kicked out of the electron transport chain. So that's forming a normal hydrogen, which forms a hydrogen. So for every 2 of those, you're going to meet up with this oxygen molecule. So remember, oxygen comes in the form of O2, but H2O forms water, and so water is going to leave your cell through osmosis. Now, so then why is oxygen important? Let's talk about that. So if oxygen wasn't here, essentially, then your hydrogens would start to build up, and if your hydrogens, I don't know why my phone keeps doing that, if your hydrogens built up here, then you would start to see that this ATP synthase wouldn't work because that gradient wouldn't be high to low because you would have equal amounts here and here. And then this electron transport chain would build up. Your pyruvates wouldn't come through because you would have a lot of them here and they wouldn't want to diffuse. So if we don't have oxygen, everything backs up and it stops this entire process. So the only thing that happens here is glycolysis. And remember, you're only getting 2 ATP from glycolysis. Otherwise, I would get 2 from glycolysis, I get 2 from Krebs cycle, and I get 32 from your electron transport chain. So that's 32, 34, 36 total versus if I didn't have oxygen available, I would stop right up here and I would only get 2 ATP. So when that happens, when I don't have oxygen to kind of make this bottom part happen, up here what starts to happen is instead of these pyruvates diffusing in, what they do is they combine back with their hydrogens and they form what we know as lactic acid. So you're going to see lactic acid build up in your muscles is actually when you don't have enough oxygen to make this stuff happen. That's where you get lactic acid buildup because you're going through anaerobic respiration within your cells.